Bom, boa tarde. Continuando com nosso curso nosso mini curso que está sendo impartido pelo professor Polperieschi no Instituto de Química. A quarta parte vai tratar sobre metabolismo de xenobióticos. Esta parte deve ser aproximadamente também de 45 a 50 minutos e quando esteja perto de terminar, eu vou colocar o formulário para a presença. Então, nestes momentos, eu vou passar a palavra para o professor Polperieschi para começar a sua apresentação. Professor.
Thank you very much for the introduction. Welcome to everybody again, and we continue this series of lectures. And this lecture will about the fate of the foreign substances, so-called xenobiotics, in the body when they get into the body. What happens to them before they develop their expected pharmacological activities. Let's see what is the fate of a drug, what is the fate of a foreign substance in the body. When we, for example, take a given amount of drug, vitamin, or chemopreventive agents, whatever, per os, I mean through our mouth, first of all, the substance should be absorbed, and this absorption typically happens in the small intestine of the gastrointestinal system. After the compound is absorbed from the small intestine, it gets into the circulation, gets into the central circulation. However, before the compounds gets into the central circulation, it gets through the liver, and afterwards from the liver goes to the heart, and it will be the heart which will pump the blood through the whole body in the absorbed drug inside. In the blood, in the plasma, better to say, the dissolved drug can form a complex with the proteins, mainly with albumins. So it means the absorbed drug in the central circulation is in equilibrium between its free form and its protein-bound form. The blood carries the compound to most tissues, and in those tissues, in those cells, when the compound finds proteins of particular proteins to which they bound and biological response develops, that proteins are called receptors, or proteins or other macromolecules are called receptors. And after the binding, we can see the biological effects. Of course, the compound gets into practically most tissues. However, not all the tissues expressed the receptors. That's why the drug's action, the foreign compound's action, is tissue or specific. Of course, the dose which is absorbed should be eliminated as well. The elimination the two main pathway of the elimination are those ones through the gastrointestinal system and through the renal system. As for the eliminated form of the compounds, either the original formula or the chemically transformed, so-called metabolized formula is excreted. The fate of any drug, any foreign compound, according to this scheme, can be divided into two different parts. One part, which involves all the processes which are involved which associated with the transport of the compounds to the receptor and elimination of the compounds, and the other part of the phase is the so-called pharmacodynamic phase, in which the biological response develops as a result of interaction of the receptors, the interactions of particular cellular macromolecules. The metabolic transformation, knowledge on the metabolic transformation is a relatively new field of research. Results of relatively new field of research. And the first book, which summarized the knowledge of that particular time, appeared in 1947. In 1947, it was Techwin Williams who summarized the information, and he was the scientist who divided the transformation of metabolic transformation of foreign substances into so-called activation phase, which he called phase 1 transformation, and inactivation phase, which called phase 2 transformations. The picture over the time slightly modified. However, the scientific literature still use the phase 1 and phase 2 transformations, which involves several different chemical reactions. Biotransformation, metabolic transformation of foreign substances, typically generates more polar water-soluble metabolites, which are readily excreted from the body. I wish to note, however, that there are exceptions in which the metabolites are less polar, with lower solubility, and sometimes the metabolites are toxic. Metabolites may still have potent biological activity, however, as I mentioned, they may have toxic properties as well. Generally applicable to metabolism of all xenobiotics as well as endogenous compounds such as steroids, vitamins, and fatty acids are also involved in the biological processes commonly known as biotransformation. As it was mentioned in the previous slide, the generally metabolic pathways of foreign substances can be divided into phase 1 and phase 2 reactions. As for the phase 1 reactions, the phase 1 reactions involves oxidative transformation, reductions, and hydrolytic reactions. These are the three types of reactions catalyzed by particular enzymes, which are called phase 1 metabolic transformation. The phase 2 transformations are conjugation reactions in which the parent substance or its phase 1 metabolite conjugated with glucuronic acid, sulfate, amino acids, as for example glycine, glutathione, and or undergo acetylation or methylation. Briefly, we will see these transformations concentrating on the phase 2 drug conjugations. In the phase 2 drug conjugations, attachments of small polar endogenous molecules such as glucuronic acid, sulfate, amino acids binds to the phase 1 metabolites or the parent drugs. Generally, the pharmacological activity and thus toxicity is attenuated, and trapping high electrophilic molecules with endogenous nucleophiles such as glutathione prevents damage to important macromolecules. That's the importance, and that's one of the most important biological function of glutathione conjugations. In general, appropriate transferase enzymes activate the transferrin group, the glucuronic acid, the sulfate, the methyl group, and the acetyl group in a coenzyme form. Let's start with the glucuronic acid conjugation, which is the most common conjugation pathway. The coenzyme, uridine diphosphate glucuronic acid, is synthesized from the corresponding phosphate, and the glucuronides are either hydrophilic and water-soluble derivatives of the parent compounds. Glucuronic acid can be linked to oxygen, nitrogen, sulfur, and carbon atoms. And based on this, we can distinguish O, N, S, or C glucuronides. As for the chemistry of the glucuronic acid conjugation, here are two examples. One of the examples is the well-known drug paracetamol, and the other one is ibuprofen. Paracetamol is a phenolic derivative, and this is the activated form of glucuronic acid activated by uridyl diphosphate. The activated form of glucuronic acid in the presence of the substrate paracetamol and the enzyme uridine glucuronic transferase results in formation of the O-glucuronide, in which the compound, the phenolic or the alcoholic compound, directly link to the oxygen atom of the anomeric carbon of the glucuronic acid. In the second type of such derivatives, not hydroxyl groups, but carboxyl groups are involved, the same mechanisms by the same enzymes, and in that reactions, the ester derivative is formed involving the same anomeric hydroxyl group of glucuronic acid. As for the details of formation of the glucuronide conjugates, you can see this is the structure of the UDP, uridine diphosphate glucuronic acid, which has got electrophilic carbon atom, I mean this anomeric carbon, and this anomeric carbon is attached by the nucleophilic oxygen of alcohols, phenols, carboxylic acids, and as a result, the uridine diphosphate leaves, and the ether glucuronide is formed. So this is a nucleophilic substitution reaction in which the configuration of the carbon, the glucuronic acid, changing from alpha configuration to beta configuration. The sulfate conjugation is less frequently than glucuronidation, and the capacity of formation of sulfate conjugation is also smaller because the glucuronic acid supply is much higher on the physiological conditions than the sulfate supply. Sulfate anions cannot directly react with phenols or alcohols, so the sulfate anions have to be activated, and the activated form of sulfate anion is adenosine phosphosulfate. This is the active form of the sulfate, and this active sulfate is further modified, phosphorylated at the 3' position of the ribose unit, and this will be the final form of the active form of sulfate ion, and this sulfate ion, and this is activated sulfate, the PAPS, in the presence of the sulfur transferase enzymes and the substrate molecules, which is paracetamol in our example again, form the sulfate conjugate. Sulfuric acid as a monocotic acid is a very strong acid, so it means that the sulfate conjugates carry a full negative charge on the physiological conditions. This is the mechanism of the sulfate conjugation in a little bit more detailed form. It's this chart describes very much the same chemistry as it was shown before in somewhat more detailed form. Those who are interested in this mechanism, I just recommend the book that is mentioned as a reference in this slide. The third phase 2 transformation is conjugation of the foreign substances with amino acids. There have been several amino acid conjugates isolated and identified. Among them, the glycine conjugates are the most common one. Glycine conjugates, glycine and other amino acids like taurine, arginine, asparagine, histidine, lysine, glutamic acid, aspartate, alanine, and serine can also be involved in such a conjugation reactions, and a variety of carboxylic acid foreign substances such as aromatic carboxylic acid, aryl acetic carboxylic acid, and even heterocyclic carboxylic acids can form these conjugates. So as you can see, as a result, in the case of benzoic acid or salicylic acid, the conjugate is acetamide. For conjugates involved in formation of an acetamide bonds, and as a result, the parent compound becomes more polar in this case. As for the mechanisms of the amino acid conjugation, it involves activation of the substrate molecule, I mean the carboxylic acid. The foreign carboxylic acid, at the expense of ATP and CoA-A, in the presence of acetyl-CoA synthesase enzyme, forms a CoA-A ester, CoA-A dioester. So the CoA-A dioesters are the active forms of the foreign carboxylic acids, and by means of a transacylase enzyme, which deprotonates the protonated nitrogen of glycine, catalyzing the reaction of reaction of this thioester with the nucleophilic amine to form the expected carboxamide derivative, in which is called, in case of the benzoic acid, is hippuric acid. The fourth one, the fourth conjugation reaction, already mentioned several times, is conjugation with glutathione. Glutathione is a tripeptide. It's composed of alanine, cysteine, and glutamine. That means that gamma glutamyl cysteinyl glycine. It has got a very special feature, namely the glutamate is linked to cysteine not through the alpha carboxyl group, however the gamma carboxyl group. That's why the glutathione tripeptide has got some very specific special stability in the body. The thiol function of glutathione, as it mentioned, is a scavenger of harmful electrophilic predator drugs, metabolites, and even radicals, and they can be involved in nucleophilic substitution reactions and the previously mentioned micro reactions as well. It has got two forms, the reduced form and the oxidized form. The oxidized form is the disulfide derivatives. In this equilibrium, however, under normal physiological conditions, are very strongly shifted towards the reduced form of glutathione. The glutathione molecule itself, as it mentioned, can readily react in a spontaneous reaction or in an enzyme catalyzed reaction. The enzyme which catalyzes such reactions are called glutathione S transferases. Can react the alkylation reaction of the cysteine SH, resulting in a thioether derivative. This thioether derivative, however, further metabolized in the body, first by means of the enzyme glutamyl transferase. The tripeptide loses the glutamate unit, and the second enzyme which acts on this metabolic transformation is the cysteinyl glycinase, which remove the glycine from the tripeptide. So, as a result, only the alkyl substituted cysteine is the remaining compound, which is eliminated from the body through the kidneys in acetylated form, and acetylation of the amino group is catalyzed by the acetylase enzyme. These final derivatives are called mercapturic acid derivatives. So it means that analyzing the urine, you can identify those foreign substances, those conjugates of foreign substances which form with glutathione and resulting in glutathione S conjugates. The transferase itself is as it can be seen from the previous lectures and from this lecture, is a very important enzyme in keeping the integrity of the cells. It has got two binding sites, one site for binding the glutathione and one other site to binding the substrate. When characterizing the glutathione S transferase activities, typically the 2,4-dinitro-chlorobenzene used as a substrate, and the kinetics of the reaction can easily followed by spectrophotometric method, and the mechanism based on the kinetics of the reactions. The mechanism is already cleared, and the catalytic effect of the glutathione transferase depends on the interaction of the cysteine-thiol function with the tyrosine phenolic hydroxyl, which increase the portion of the deprotonated form of glutathione, resulting in higher amount of the negatively charged cysteine-thiol, which shows much higher reactivity towards the electrophilic centers. Here is an example of glutathione conjugation, attacrinic acid. It was mentioned in the introduction. It is a diuretics. This attacrinic acid metabolism is the main pathway of attacrinic acid metabolism is represented by its GSH transferase. The GSH conjugation of the attacrinic acid were investigated in details, and because as a result of the addition reaction, two chiral carbon centers are developing in attacrinic acid. The stereochemistry of the different glutathione S transferase enzymes were also could be investigated. It was found that the glutathione S transferase 1 form results in formation only one of the possible diastereomers, which is the consequence of the selective protonation of the enolic intermediates. We also investigated the glutathione addition onto amine molecules in cooperation with professors from the Guyana chemistry department of the Federal University of Goiás, and we, among others, compared the reactivity and stereochemistry of two chalcone compounds. One of them was a simple hydroxylated derivatives on the H ring, and its analog, which was the bis-Mannich derivatives of them, and we found that in case of the bis-Mannich derivative, one of the possible diastereomeric pairs formed in a much higher amount than the other. So diastereoselectivity were observed in the reaction, and this diastereoselectivity was presumed to be associated with a cyclic intermediate, and the preferred conformation of this six-membered cyclic intermediate governed by the preferred position of the bulky aromatic substituents was considered to be the reason for the diastereoselectivity of the addition reactions. Going further on the different types of conjugation reactions, the next one is the acetyl conjugation, or briefly the acetylation. Acetylation is a very common transformation, not only exogenous, however, for endogenous compounds as well, which results in acetyl derivatives of amines or alcohols or phenols in most cases. As for the foreign substances, any drug, any xenobiotic with aliphatic or aromatic amino groups, amino acids, sulfonamides, hydrazine, and hydroxides can form acetyl derivatives, and the enzyme which catalyzing these reactions are the so-called N-acetyltransferase enzymes, and the co-substrate of these enzymes is acetyl-CoA. So acetyl-CoA is the co-substrate. In this case, aminobenzoic acid is the substrate, resulting in the acetylated derivatives. It should be mentioned here that acetylation reduces the water solubility because instead of a relatively polar amino group, a less polar acidamide group is formed. So in this case, any drug, for example, in case of sulfonamides, that undergo acetyl conjugation, it should be taken into consideration that the solubility is decreasing. That's why medical doctors recommend for those people who take sulfonamide drugs to drink a higher volume of water than general to prevent crystallization of the less soluble acetyl derivatives in the kidney. Acetyl conjugation is, as I mentioned, it's also a nucleophilic reaction of the attack on the nucleophilic nitrogen or nucleophilic oxygen on the positively polarized carbon of acetyl-CoA, and as a result, resulting in the acetylamide derivatives. The next one, the last one, but however not the least important conjugation reaction is the methyl conjugation. It's a minor conjugation pathway of xenobiotics. However, it's rather important in biosynthesis of plenty of endogenous compounds. For example, norepinephrine, dopamine, serotonin, histamine, of which methyl conjugation modulates their bioactivity of the compounds. A variety of methyltransferases, such as catechol O-methyltransferase, phenyl O-methyltransferase, N-methyltransferase, S-methyltransferase, and so on, are responsible for catalyzing the transfer of a methyl group from the S-methyl adenosine co-substrate, which is synthesized by the S-adenosine methionine synthetase. This is methionine, this is adenosine, and this is the activated form of the methyl group, and the methyltransferase can transfer this activated methyl group to the nucleophilic site of the target. For example, here is norepinephrine as an endogenous compound, and here is the S-adenosyl methionine, adenine, ribose, the methionine unit with the activated methyl group, and this activated methyl group, as known from basic biochemistry, can be transferred to an oxygen atom of norepinephrine by means of the catechol-O-methyltransferase, or the nitrogen atom of norepinephrine to the nitrogen methyltransferase and converting norepinephrine to epinephrine and methylating the primary amino group of norepinephrine to a secondary amino derivative. Let's see now a particular example. What happens when a concrete, a particular flavonoid, for example, quercetin, gets into the body through the mouse. Quercetin, in its natural forms, exists as glucuronides. The aglycone, the quercetin aglycone, is linked to sugar units. And this is the form quercetin gets into the gastrointestinal system. Such form is two polar, and these polar forms cannot be absorbed by the small intestine. However, there are several hydrolytic enzymes which hydrolyzes these glycosides, removing the glucose units from the aglycone and resulting in the small organic molecule quercetin. Quercetin can get into the intestine, the epithelial cells of the intestine, by a very special functionality which is called sodium glucose transporter into these enterocytes. Okay, so in the enterocytes, the hydrolytic transformation occurs, and inside the enterocytes, the quercetin, now we are already familiar with the different metabolic transformations. The phenolic hydroxyl groups and the enolic hydroxyl groups are involved, and partly by interaction with the sulfotransferase enzymes, the sulfate derivatives is formed, and by means of the glucuronic acid transferase, the glucuronic acid derivatives is formed of the quercetin. Glucuronic acid, as you can see, carries a carboxyl group which is partly ionized, so it means the polarity of the compound, the aglycone, increases very well and very substantially as a result of this conjugation. This conjugation is these derivatives are formed in the enterocytes and partly in the enterocytes and partly in the hepatocytes. The hepatocytes have got higher capacity to catalyze these reactions, and in the plasma acts as a catechol O methyltransferase, which can methylate the meta hydroxyl group of the aromatic substituent. So this was a particular example how a foreign compound undergo metabolic transformation when it gets into the body. The non-hydroxylated chalcones, metabolic transformations on the non-hydroxylated chalcones, we have only limited data. One of them is those results mentioned in these slides. As you can see, particular cytochrome P450 enzymes can catalyze hydroxylated oxidation reactions of the chalcone molecule, resulting in four hydroxyl derivatives, four prime hydroxyl derivatives, and two hydroxy derivatives of the parent chalcones, which afterwards can, of course, further undergo further metabolic transformation. For example, conjugation with sulfate or conjugation with glucuronic acid. Finally, I will wish to show the result of an in vivo investigations of metabolism and absorption and metabolism of two chalcone derivatives previously mentioned. This experimental protocol uses experimental rats, which are are from which the food was withdrawn before 24 hours of the beginning of the experiment. Afterward, they anesthetized, and we got a median laparotomy of the animals, and we isolating roughly about 10 centimeter length segment of the jejunum, which is the upper part of the small intestine. We have a pump and we have a reservoir in which we dissolve the compound of interest, and by means of a peristaltic pump, we pumping through the compounds of interest of this 10 centimeter length small intestine unit in so-called recirculation modes. The perfusion is running for 90 minutes, and over the 90 minutes, we collect 14 samples, and the obtained samples were analyzed by validated HPLC UV and HPLC fluorescence detection. The two compounds, you see, are those ones which previously mentioned before. One hydroxylated compound and a bismanic analog of the compound. What we could see that the relatively apolar non-Mannich chalcone derivatives is fairly fast absorbs from the perfusion medium. It means the absorption of the compound is fairly effective in comparison with the Mannich derivatives, which under physiological conditions are deprotonated, and which means that the protonated Mannich derivatives, the absorption of the protonated Mannich derivatives seems to be, showed to be less preferred than the non-Mannich derivative, than the non-charged Mannich derivatives. However, at the slower rate, these compounds were also absorbed from the small intestine. While analyzing the the samples, the intestinal samples. We were identified by mass spectrometric derivatization the sulfate derivative of the non-Männlich chalcone, the glucuronic acid derivative of the non-Männlich chalcone, and the glutathione conjugate of it. And we were able to identify the same kind of derivatives in case of the Männlich chalcones too. It means that the intestinal metabolic fate of the the chalcone derivatives involves the sulfate and glucuronic acid conjugation involving the phenolic hydroxyl groups and glutathione conjugation involving the polar carbon-carbon double bond as it was referred to us as a Männlich reaction of the glutathione with the chalcones. Briefly, that's about the feature of foreign substances in the body with examples with two particular compounds, quercetin as an example of flavonoids, and two chalcone derivatives. One of them is a phenolic one, and the other one with the phenolic chalcone with bis-Männlich functionalization. Again, thank you very much for your attention.
¿Alguna pregunta para el profesor Paul? Well, professor, no tenemos, we don't know, we doesn't have, the people doesn't have any question. No question. Amanhã, lembrar que amanhã às 14 horas volvemos com a última parte do minicurso, que será também dividido em duas partes. Às 2 horas da primeira parte, 15 ou 20 minutos de intervalo, e depois continuação a segunda parte e final. Muito obrigada por assistirem vocês ao minicurso e espero ver vocês amanhã. Tudo bom? Até amanhã. Andy.